All right. Well, we come to the end.
The end?
I think we were scheduled to have about a 1-hour discussion. However, I think very profitably the last sessions ran a little longer. We started a little later, and I wouldn't have changed any of it. And David Glazer did actually fit it in, which was very good with 56 slides, which is possibly an ICDA record at this point. Several people I know have flights, and so we're going to end at 3:45, one of those people being Celia. And so that's going to enforce a hard stop at 3:45. But we have a little bit of time for the next 20 minutes to just open the lens to the kinds of projects, the kinds of plans, the kinds of policies, the kinds of recommendations we want to make about those in the first version of the white paper that we plan to have done by January. We haven't specified whether it's January 1st or the end of January. That's the experimental error in the system, but no later than January. Oh, sorry, we have specified 2020 though. Yes, Mark is keeping us honest that we could always just, if we miss that deadline, go to 2020. No, we January 2020. So we wanted to throw it open for a little bit of discussion, just to help us think more about concrete projects, more to make sure we had a common sense of it. So just to warm us up, there's been some discussion of, shouldn't we know all the enhancer-promoter connections in all cell types? Yes. Now, how should that happen? What sort of things might need to happen to make that possible? Are there pilots that would show us that we really could do that? Like, how would we find them? How would we prove that we had them? Could we start predicting them and showing our predictions were satisfied by experiment? There's a lot of thinking about such a project. We're not going to get to the level of writing a grant proposal, and it's not anybody individual's project. We're trying to define a good project that would find such a thing. Understanding the consequence of all common human variance with respect to immediate transcriptional effects in a cell, say all these EQTLs that Thule says we sort of have for tissues. Can we get down to cell types on it? What do we need to know about it? Those are examples of things we might choose to say are important to start down the path for now. Maybe not. I'm just using this as warm-up here. We might sort of be thinking of a time frame not of what we can do in a year, but neither what we could do in a decade. A decade's kind of long for conversations like this. I think the kind of things we might be thinking about, what could get done in the next 3 to 4 years, maybe 5 years, something like that. So serious work, well-organized work. These machine learning things, well, we've got them running in a lot of different labs, but really making those things that people can beat up on and say, wait, do I believe it? Anshul said, many of these things work very well in sample, but not out of sample. Well, it's, once you turn the harsh spotlight on it, we'll begin to see where is it not working and how it'll push the field to make it work. So there could be a set of challenge problems. Well-define a set of challenge problems for which, for example, some data sets might have to be created and put those challenge problems out into the field. Now, funders might say, oh, we want to fund those. We want to make sure that labs could apply for a little bit of funding to help support a student working on the challenge problem, or they might decide something else. Our job isn't to work out a funding mechanism. Our job is to work out what are good, meaty, well-organized kinds of problems for us to take on. Summary statistics. It was said that we should urge everybody to put their summary statistics in the GWAS databases. Yeah, maybe we should do more than urge. Maybe we should get to the point of really trying to push that this be a requirement of publication in all leading journals. So we might propose here not a challenge problem, but a policy, and figure out what does that mean. But that would mean people would sit down and say, is that practical? Are there problems with that? And try to think through a well thought out policy. We've had conversations about Africa, 2 million people in Africa. Well, there's no chance that by January we'll organize the exact right thing, but we could scope it out well enough that a regional working group that also involved people from around the world, as well as leadership from the region, could begin to develop such a thing. So I paint this to say there are a wide range of things that are concrete, they differ in character, but they might move the field forward a lot, and they're doable things. That's the kind of thing we're hoping for. They can range from new methodologies. Hillary was talking about new methodologies. So I'd love to throw it open, but I wanted to give those as some examples of the range of things that might be in bounds. And I think the goal over the coming several months is, with the work of the working groups, to have people try to write half-pagers, saying, here's something that might be a good idea, and share them in such a way that others can weigh in and say, I have no idea what you're talking about, or this sounds like a good idea, but it could be improved in some other way, or have you thought of this? But to have a real conversation. Again, if it can be done with existing grant proposals, if you can write an R01 on it, then write the R01. We don't need ICDA for that. It's to look at those things where there's a point to bringing together a community to work on it. That's sort of my starting point. Do you want to add to that or charge them in some way? And then we should throw it open to them.
So I think one of the things that really excited me was 2 comments that Thule and Rory made. So the first one, should we make another GTEx? It sounds exhausting, and I think it sounds exciting. If we think we're going to expand diversity, just tying on the point that Hillary made, we should probably do another GTEx, probably in Africa, probably in South America, and maybe in India, where the 3 places that were mentioned, and we probably should do it on a single cell base level, so we expand what we know. Those are huge projects, maybe not within a year or 2. But laying the foundation for it and expanding because it's the right scientific effort, I think would be exciting. So I think when people think about scope, one shouldn't be limited about it, but what's the best thing to do? And the thing that Rory said, which I do agree, is a vacancy in the white paper at the moment. It's actually, nobody's proffered any insights to what we're doing with the layers of proteomics and so forth. And I really think, having seen Kari's decode example and so forth, that there is a lot left to be done there, and those are huge projects and probably really expensive. I don't know if you do the proteome in biobanks across the planet on a proper scale, if that's an R1, but I would assume not. So I think there is scope in thinking about it. So some people have already started to email with proposals, or, I would want to lead an effort around software and data technology. Somebody had emailed Go You. That sounded really exciting. And I think, again, I just want to encourage people to think about the ideas they want to put forward. And the working group leaders, Eric and I and Rachel, will help facilitate the discussion around that.
Yeah, and when we mean put forward a proposal, just again, it's not that you would do it. It's not you're making a grant proposal. It's that it should be done. Your mention of Rory reminded me of his point that maybe we should think about not making more UK biobanks, but what are the next innovation in it? Like maybe using medical systems. Well, how do we turn a good hospital into a biobank? And maybe we can create an incentive for really good medical systems to step forward. And who knows, maybe funders will even fund them to do the demonstration projects to turn those into biobanks. And who knows, maybe in some of the regions of the world that'll be better than copying exactly what was done.
But also iterating what we have said, we do not want to duplicate or compete with ongoing efforts.
Yeah.
I think that's really key. We're not going to duplicate or compete with the Human Cell Atlas or the 100,000 cohort. It's not the intention at all. I think we want to augment, facilitate, and collaborate.
That's right. And we can improve some of the things we've heard about today. David Glazier's talking about this Terra platform. People should say, well, how come it doesn't do X, Y, Z, and W? We need to ensure that it covers other things. You know, I think they are working to make single cell biology available in the same context, but I think particularly as the analytical people ask, is it really going to serve our needs? They should lay out what those needs are and be demanding of it. And of course, we want multiple open source platforms that do things. But anyway, so I think we've said enough that maybe juices have started flowing and people should jump up.
Ewan is already up at the microphone, so.
So Ewan has overcome his shyness and is going to fire away.
It's been tough. I've taken a lifetime to overcome my shyness. Actually, so
Manolis has overcome his shyness.
One thought to this, which I'd be interested in both your thoughts on, is that there are an awful lot of human diseases that we care about. The phrase common disease encompasses a very large number. And I think it's very, very easy for people to look at what has happened in Crohn's disease or type 2 diabetes and assume that because someone has done it there, that it's somehow just a click on David Glazier's amazing system to do the same thing. And I actually think it'd be a good thought exercise to ask, how many analysts years do we need over the forthcoming decade to do every common disease that is at least 2% frequency in the OECD? I'm making up the criteria, but some practical criteria and some reasonable assumption of how long an analyst takes to do each disease, some idea of how you could batch it up, because I think laying out some of the scale of the problem is hopeful. And I also want to emphasize the fact that we can do it in type 2 diabetes and Crohn's does not mean it's like a snap your fingers for all other diseases.
So just two quick comments before I hand over to Eric. So the first one is that when I give a talk around this, I usually show a slide from Giant that is a Manhattan plot, and people get bored and they think I'm from the Big Data Institute, and they're like, why are you showing this if this is a GWAS slide? And that single one slide represents over 36 years of collected manpower of analytics in terms of data generation, QC, which I think you can spend a lifetime on, and then the singular analyses and just iterating that, which is incredibly time consuming and boring. So when somebody comes to me and asks if I can run a quick analysis, the answer is no. So I think, we have enough collected experience of going through that in this room and also in the community for people that are watching, that I think it's a really valid point you're making. So I don't think we should underappreciate it, but I'm hoping that we as a community have learned that lesson. So, the tools that David and Helen and others are putting forward should not be seen as a shortcut for somebody mediocre to just click around. I think it should be viewed as something that will facilitate collaboration around the world, so we can come together in a sensible way. The second point I'm going to make before you jump up again is that I had a great discussion with Dr. Bianchi before today. So there are traits and diseases where it's almost impossible to receive funding. I have a particular interest in reproductive and women's health, and it's just almost impossible to get grant funding for that. So with these biobank data, we have now managed to do, GWAS for recurrent miscarriage. It's never been done before. We have run a GWAS on some other traits and pregnancy complications, and it's a treasure trove for those traits and disorders. And it gives an opportunity for underrepresented traits, not only scientists in places on the planet where it's difficult to do it, but also actually propelling serious biology and addressing disorders that would just never be prioritized otherwise.
Yeah. Just going through this paper exercise of trying to work out the number of analyst hours,
yeah.
What you just mentioned there about QC and kind of, all of these very complicated choices, which covariates do I regress, which ones do I leave in, how do I decide on all these things? Now, I think we can do faster than we've done it before, but I just don't think it's going to go to zero, anywhere close to zero.
It shouldn't go to zero.
It shouldn't go to zero. It's
It
very hard.
mustn't go to zero.
So I think being responsible about trying to describe the amount of analyst resource we need
Yes.
would be an incredibly useful paper exercise. Yeah.
I agree with you completely.
I'm looking out for your flight.
Yeah, okay.
So-
I got excited.
No.
Thanks.
Ellie, yeah.
Yeah, Ellie Zaghini from the Helmholtz Center in Munich. So I found this meeting really inspiring and great, and I think it's fantastic that we are now starting to think about doable projects, but also starting to plan bigger things where we can lay the foundations for those to happen. But I do wonder if we're perhaps missing a bit of an opportunity with the collective brain power in the room. Are we really engaging with colleagues outside of our immediate discipline? Should we be talking to engineers? Should we be looking at technological advances that have historically really catalyzed progress in our field and asking how can we solve this issue of scale that Ewan poses, or the issue of proteomics being too expensive or spatial transcriptomics not being quite there yet? Should we be making recommendations to that community as to what we need to really accelerate progress?
I think the answer is yes, and that's exactly what we're trying to do. So we should engage all facets of community, and we won't be able to capture everyone in the first couple of meetings we have, but we are open to, if we have missed any parts. We have a quite diverse group, I would say, from age range to diversity on different axes and to stakeholders and so forth. But I agree with you, there is always the chance that you miss out on parts of the community.
Manolis is going to demonstrate his capacity for brevity.
Okay. Already laughing. I just want to sort of reiterate the point that Tuli made earlier about a new GTEx. And in fact, we should not be thinking of that as just a genetic exercise, a genetic diversity exercise, but actually an environmental diversity exercise. So you can think of it from the point of view of the different exposures of the environment and how it interacts with the genetic effects. We can think of the diversity of microbiomes in the different tissues, and actually can teach us a lot about so many of the things that we're talking about. Either we do it at the single cell level or we do it at the tissue level, as we've been doing. And in fact, it so happens that about 3 or 4 years ago, maybe more, with Kristin Ardley, we had a meeting in Barcelona with about 15, 20 people that were working at GTEx at the time. I think you were there too, right? And we discussed exactly that. And at that time, of course, it seemed like a crazy idea. But we do have the notes for that meeting. We never actually got to write the paper, but we do have the notes. So Kristin and I could sort of bring back together and give you something more at a half-pager, but something that is probably a little bit more organized with deep ideas from that meeting.
All in favor? Aye.
I also think we can
When do we get
add
them?
tissue proteomics and all kinds of stuff.
Thank you.
Jesse.
So the Human Cell Atlas in the next 5 years, we imagine, will have single-cell RNA-seq and probably single-cell attack-seq in many cell types. And if we imagine expanding across populations and environments, we're going to have a readout of what our cells look like in many genetic, environmental contexts in many tissues in the body. And with that data, I think we have the opportunity to start thinking comprehensively about answering a few questions, like predicting the effects of sequence variants on regulatory activity and gene expression, like Anshul talked about. Those data, in theory, can help us connect enhancers to their target genes. If we learn computational models, leveraging CRISPR data and other things, in order to learn how to use those data sets in every cell type to make those connections. We can learn how to predict which genes affect which programs in cells, let's say as represented in gene expression. And so I think one overarching challenge for this group to think about is how we can build truly comprehensive maps of every cell type and state given some measurement in a person to connect variants to their molecular functions.
Sounds super exciting. I would also really want to know the definition and have a full maps of the pathways in every cell, because I personally don't know what pathways is. I'm certain a lot of people feel they know, and I'd be so excited if you told me. I've been tweeting with you and about it, but I still don't know.
Or we take two quick last questions. Vigil?
I think one thing that's important is for the success of all of these projects, you need to have the trust of the public. And earning the trust of the public right now is tricky. But one thing that you can do is to write for the public. Write about your science simply and clearly for the lay public. And there is an avenue for you to do that. Little self-promotional here. The Conversation, we deal with scholars from universities. You work with editors directly, and we get your stories out there in your writing, in your language. And I can tell you that it goes a long way to educating the public. So I will be reaching out to you soon. So I hope you'll take part.
Thank you.
Last.
Yeah, the disease session absolutely blew me away in terms of complexity and potential. At the same time, I thought if we're moving towards genomic medicine, integrating genetic testing and research in the patient community in their particular context for their particular disease, we could create actual cohorts, incremental population cohorts over time. So move away from sort of the original, UK Biobank, all of us model populational, and create patient, real, virtual incremental cohorts that'll give us more contextual data to then use the population one as controls and then the disease as actual reality.
And that ties into what Kari said as well, that if we're building new cohorts, maybe what we should do is actually be in healthcare.
Yes, that's exactly it.
Yeah.
All right, we have promised to close. I will say a word, you will say a word. The word I'm going to say was amazing. The talks were really amazing. This was one of the best meetings I have been to in a very long time. I think the quality was enormously high, and I want to thank all of the speakers who gave overview talks, deep dives into things. So if we can please just thank all the speakers. And we took the first step to really building a community. There's a tremendous amount of work to go. Building a community is a lot of work and takes a lot of tending of the garden, but thank you everybody for coming out and being part of it, and to the 300 or who knows by now how many thousand people are watching on the web. So Cecilia, close us out.
I'm just going to say thank you. I'm going to say super amazing because I like to sort of add words. I'm going to thank especially Rachel and Mina and Amy for being fantastic, and also to Caroline again for co-organizing, and to NIH for hosting because it's been amazing. I want to thank everyone not only for great science but also for the effort and time you're investing in coming here. It's not something we take for granted, but it's something we cherish. So we're really grateful for that. And I think we're in such an amazing opportunity to really make a dent on the underlying biology and sort of propel it forward. So we should really leap on the opportunity, be excited about it. And I'm looking forward to do it together. And thank you, Eric.
We'll be in touch soon.
Hey.